Good morning everyone. Today I am presenting case number 12. That is Plasmodium vivax. It has more than 100 species which is present in both animals and humans. It has different variants like Plasmodium vivax, Plasmodium ovale, Plasmodium malariae and Plasmodium falciparum which are mainly affects the humans. Coming to geographical distribution, vivax and falciparum more common than the ovale and malaria. More than 200 million people worldwide are affected and more than 1 million deaths per year occurs. That's why most common lethal infectious disease. Habits. It is mostly transmitted by female anaphylaxis, which is by their sexual cycle, and in the humans, liver and RBCs. This is transmitted by a sexual cycle. RBC age variable. Vivax affects in the youngest erythrocytes, malaria affects oldest erythrocytes, and falciparum affects every age of the RBC. Morphology. Peripheral blood stained with Leishman stain. Tall, small trophozoites which are also called as immature or ring forms. Infected RBCs, first they will form a ring-like structure. These are dot or rod-shaped nucleus that is red color, and there is a peripheral rim of cytoplasm which is bluish color. Central clear vacuole-like area which is not stained. Large trophomers, these are ring forms and these contain fine grain-like pigment hematine. Next stage is seasons. These large trophozoites are converted into seasons and these are nuclear cytoplasmic fragments, and these are later converted into merocytes. Gametocytes, these are two forms, male and female, which is indistinguishable. These are fully grown and these are rounded and which occupies most of the RBCs. Falciparum is sausage shaped and crescent in the RBCs. Others are round. You can see here plasmodia in the RBCs, which are in ring forms and gametocyte forms. Life cycle host. Definitive host is female Anopheles. Intermediate host is man. Vector is female Anopheles. First sexual cycle initiated in the humans, then these converted into gametocytes which is present in RBCs, and then it will transfer into the mosquitoes and fusion of male and female gametes occurs, then oocyst is formed and many sporozoites or sporogony are formed. Sexual cycle is sporogony or sporozoites are formed. In a sexual cycle, schizogony, that is schizones are formed. This is the life cycle of the plasmodium. First, stage 1, the mosquito takes a blood meal, then it transfers to the liver cell. In the liver, human liver stage, the infected liver produce more schizones. Then these schizones rupture and then come into the human blood stage. Later, these are formed immature trophocytes or ring stage. Then these converted into mature trophocytes or gametocytes. These mature trophocytes are converted into seasons and seasons get ruptured and again they will go into the human blood stage and immature trophocytes. Again these gametocytes, these will taken by the mosquitoes, and in the mosquito macro gametocytes and converted into the microgametes, then ookinete and then oocyst and mosquito stage, and this oocyst is ruptured and released into the bloodstream by the mosquito bite. This is the life cycle of the malaria parasite. Pathogenesis. Usual incubation period Vivax is 14 days, malaria 28 days, falciform 11 days. Transmission by the mosquito bite, IV drug abuse, blood transfusion, transplantation, that is congenital, and is associated with fever, anemia, and splenomegaly. Malarial relapses. Plasmodium vivax is 2 years, para erythrocytic stage, then liver parenchyma, then they dormant but viable. When the resistance is lowered, these para-erythrostatic stage, they will get activated and they enter into the erythrocytic cycle. But not in Plasmodium falciform, as no para-erythrocytic stage is present. So, transmission other than mosquito bites, there is no relapses. Natural protection is seen in sickle cell trait individuals and Duffy blood group antigen negative individuals and glucose 6 phosphatase dehydrogenase deficient individuals. Premutation, partial immunity, it is after infection. Humoral antibodies, like these block merocytes from the invading RPCs, and low level of the parasitemia, that is only low grade symptoms can occur. Commonly involved organs: blood, spleen, liver, and bone marrow. Signs and symptoms, that is abrupt fever with chills and rigors, headache, initially may be continuation then periodic. Fever may go up to 41 degrees Celsius, along with nausea, vomiting, pain, anorexia, drenching sweats afterwards. Then he will feel well between febrile episodes. There is also splenomegaly, and 1/3 cases we can see hepatomegaly, anemia, falciform fatal because of brain and kidney damage. Blood examination, microscopic examination, it should be drawn during pyrexia period, not after even single dose of antimalarial. Thick and thin smears are made, and these are dried and stained. Thick smear to identify the presence of organism, and thin smear to identification of the specific species. Thin smear, we can take a single drop of blood and we allow it to spread on the single cell layer, and it is stained with Leishman stain and seen under the oil immersion lens. Ring shaped strophocytes in the RBCs are seen. Falciform gametocytes seen as banana or sausage or crescent shaped. Other species gametocytes are spherical in shape. If more than 5% of RBCs are infected, and diagnosis of falciform is made. Thick smear, for this 3 to 5 drops on the slides are allowed to dry. Several cell layers thick layers are made, and it is stained with either gyms or fields and seen under the oil immersion lens. Stain usually removes HB from the RBCs. This is thick smear and thin smear. Total leukocytic count and differential leukocytic count. Usually there is leukopenia and monocytosis containing pigments are seen. Bone marrow and liver biopsy can be done in difficulty cases, and therapeutic tests like antimalarials given if fever subsides. Diagnosis is made. Serological tests like fluorescent antibody testing, complement fixation test, flocculation test, and hemagglutination tests are also done. Coming to treatment, falciform easily treated before complications because there is no relapse or no para-erythrocytic stages found. Chloroquine is treatment of choice for sensitive strains of the plasmodia, along with primaquine, mifloquine, or quinine and hydroxycycline are also used. Prevention by chemoprophylaxis, mosquito netting, window screens, mosquito repellents, protective clothing, special care during night time. These are all some preventive measures. Thank you.